Hi, my name is Tung, and today I'm going to talk about MATLAB in bioinformatics. So, bioinformatics is the development and application of computational tools in managing and understanding all kinds of biological data. It involves the technology that uses computers for storage, retrieval, manipulation, and distribution of information related to biological macromolecules such as DNA, RNA, and protein sequences, as well as their structures and functions. So, overview, we talked about MATLAB applications in bioinformatics and about bioinformatics toolbox of math work, and show that MATLAB is very easy to integrate and deploy bioinformatics tools with MATLAB. And last part would be one example of how to use MATLAB and bioinformatics toolbox in order to classify some biological data. So MATLAB is able to uh, MATLAB provides tools to do sequence analysis, such as aligning pairs of genomes of genes or comparing the whole genomes, constructing scoring matrix, and using that matrix to measure the evolutionary distance, and from that a distance matrix is constructed in order to build a phylogenetic tree, and with this phylogenetic tree we could infer the evolutionary relationship between biological data. And MATLAB also provides tools to uh, for visualization of microarray data or for gene expression, and it also provides tools and functions to study genomics and proteomics as well. So the bioinformatics toolbox provides function to retrieve genes, genomes, and proteins from GenBank or NCBI, EMBL. It also the toolbox also provide function to convert DNA sequences to proteins or to amino acid sequences. Um, it provides algorithm and to do perform sequence analysis, um, including global alignment and local alignment, and it provides tools to for protein visualization and perform cluster analysis as well. So um, bioinformaticians get pulled in two directions. Biologists go to them and ask them to analyze the biological data. On the other side, the bioinformaticians need to create the prototypes and algorithms for the software engineers to implement. In more details, researchers in the biotech field are using many different programming languages such as C, C++, Java, Perl, Visual Basic, SQL, R, SAS, and the bioinformatician have to try to integrate them, since having one tool to do all analysis is a big time saver. However, integrating tools from various programming languages is difficult, given that closed source tools are not customizable. So MATLAB is able to do so easily. Moreover, deployment is key. Bioinformaticians spend a lot of time doing analysis for biologists. If they instead could develop a tool and deploy it to the biologist, then the biologist could do some of the analysis by themselves and thus save some time for both sides. So with MATLAB's deployment tools and its UI design environment, the bioinformaticians can easily create the GUIs and analyzes that the biologists want, and they can also create the prototypes that the software engineers request. Doing this in one environment saves a lot of time. So this is an example of using MATLAB and bioinformatics toolbox in order to classify some biological data. So in this example, we're gonna download the genomes using GetGeneBank and FastRide. So GetGeneBank would help download the data from the website, and FastRide, using FastRide, we're gonna just write all the data into a nice format to our computer, and we don't use those data to perform cluster analysis. So the method in this example is like given a DNA sequence, there's associated binary indicator sequences, and mathematical moments are constructed using discrete Fourier transform, and with these moments, we're gonna construct a distant matrix in order to perform cluster analysis, and with that we're gonna get the phylogenetic tree using the sequence linkage from a function from bioinformatics toolbox. So this is one example of the tree. This is Mammo's data. As you can see, the data is classified correctly. For example, this one is the primates group, and this is prudential group, and this is carnivore group. So this is the demo code. So this is the data that we want to download. These are accession number and corresponding abbreviation name for that accession, for that genome. So we use this GetGeneBank function. This will help download the data from the website, and here we are writing all of these data into a FASTA file. So this is the main function of the code. This is a function that help to construct moment vectors, and here the distant matrix is constructed, and this is sequence linkage function from bioinformatics toolbox. This will help to draw the phylogenetic tree, and we can infer the evolutionary uh, from there of the data from there. So the main function, we hit run, and we have options to we have many data sets to choose. Let's say we start with mammals, and get a tree for mammals, which is similar to the tree we had in the slide. This is influenza A virus. So you see this is group H1N1 and group H5N1, and the rest are classified correctly, but H1N1 and H5N1 are common, and I think you all know about this. We also have coronavirus. Coronavirus. So you see group 4 here. So there are like 5 groups, but group 4 here is severe acute respiratory syndrome, or SARS, that I think you also all know about. Okay, so this is demo. Can hit quit and close the menu. I think that's it. References, and thank you for watching.